My name is Kwabena, Badu-Nkansah. I work in a Heinz lab, and what we do is we work on angiogenesis. Angiogenesis is the process in which blood vessels are formed. You see this happen various times throughout development, and in this case, in pathogenesis in cancer. One thing that's really important is to know, how does this happen? How can tumor cell or cancer cells be able to send signals to attract blood vessels and be able to incorporate it into their own system? So one of the things that we're interested in looking at a class of surface receptors called integrins, and what they do is they reside on the outside, the periphery of a cell, and are able to interact with the cell's extracellular environment and send cues from that, based on that, to the interior of the cell, where the cell is able to respond afterwards. And so what we do is we knock out genes that code for these proteins and see what happens. We have certain agents that will bind to different different types of cells. And these agents will fluoresce. So we have one that binds to endothelial cells, and we have one that binds to a class of cells called Merle cells. And these cells are cells that surround blood vessels, and their role is to maintain the integrity of blood vessels. And so it's a nice way of seeing whether these cells cover blood vessels differently or whether they maintain integrity differently once we've messed around with the way they could sense their outside environment. We use a method called confocal microscopy to take this picture. What confocal microscopy does is sends light through a very small aperture. And so what that allows you to do is basically filter out all the out of focus light. And the result is a higher resolution image. And so what you're able to couple with this is the ability to move this actual stage your specimen sits on. And so you can move it up and down, and as a result, be able to get an image from different levels in your sample. And once you put them together, you get a 3D sort of rendering.